Okay, so we'll continue on. We went through the intro. We talked in general about water-soluble vitamins. And remember, any of that stuff is fair game for the quiz. The stuff on DRIs, you want to go back through that, make sure you understand those details. For structures, I mean, this is a structure of thiamine, and I said you need to be able to identify this. So for example, if I have the structure of thiamine on there, you need to know that it's thiamine instead of like thiamine pyrophosphate or thiamine triphosphate. Now I'm going to make you draw the structure. We talked about the sources, relative amounts in different foods. The RDA is the DRI. We went through the reaction, and again, I won't make you draw the structures for this, but you need to explain this process, the bridge reaction process, and how thiamine is involved. We talked about maple syrup urine disease. And as we're going through these major functions of thiamine, we're now going to drop down to the second major bullet point that says the synthesis of pentoses and NADPH. This is as a coenzyme as thiamine pyrophosphate. So the principle is the same. The thiamine is modified to a thiamine pyrophosphate, and then it can bind to an enzyme and allow that enzyme to function. So in this case, the enzyme is Trans-Ketolase. And this diagram is not in your textbook, and this is actually, it's not a complete diagram. There's some steps missing, but this I think will give us the basic overview of what's happening here. Now, it says Trans-Ketolase and the hexose monophosphate shunt. That's what HMP stands for. Hexose monophosphate shunt. It's also known as the pentose phosphate pathway. You might have heard about that in other classes. So here's a basic overview of how it works. And in 328, I think one of the things on the diagrams was it went like this. Or if you might have seen this in biochemistry. You have, for example, glucose converting to glucose 6-phosphate. If you're going through glycolysis, it does this. Another option for glucose and glucose 6-phosphate is the hexose monophosphate shunt. Basically what cells do is they take glucose and its carbons and they convert it to hexose phosphates and pentose phosphates. Okay, so what's happening is, let's look down this oxidative side first. And you don't have to regenerate this, but in general, what's happening? Glucose 6-phosphate is oxidized. As it's oxidized, the hydrogens are added to NADP. Not NAD, NADP. Okay, so it becomes NADPH plus a proton. So you have 2 electrons that are now part of this molecule. Okay. Then it's rearranged, we go down another couple of reactions, and it's oxidized again. Another molecule is oxidized. So the glucose 6-phosphate starts kind of an oxidative series of reactions. So you end up with these molecules. NADPH is used for biosynthetic reactions. NADPH is used for biosynthetic reactions. Okay, that's why it's different than NAD. When you say NAD, what do we think of? Energy production, right? Even at 7:45 in the morning, we think energy production. NADP, biosynthesis. NAD, energy.
Can we also say that's broader? NAD is for metabolic reactions, and NADP is for anabolic overall, or should we not associate that?
That's fine, yeah. That's another kind of layer to it, yeah. So now what happens is this,
What was the second thing getting oxidized again? Sorry, is that another G6P?
Here,
Something got oxidized the second time. What was that?
this is 6-phosphogluconate.
Okay.
Now, here's one of the important parts of this. Look what you end up with. You end up with a ribulose 5-phosphate. In other words, a ribose phosphate. Okay, that's converted to another form. So now these right here are also ribose phosphates. These can be used to make DNA and RNA. Okay. Now, this reaction, these reactions are part of a cycle. So if these molecules, these ribose phosphates, are not used for nucleic acids, they can basically convert back to fructose 6-phosphate and go back through this cycle.
If they're not used to make nucleic acids, then what?
They can cycle through the previous reactions essentially. So then what are you doing? Well, you're kind of making more of these. And these are important for biosynthetic reactions. So let's say fatty acid synthesis. Remember that when you drew out that pathway for fatty acids on your poster and we went through it in lecture? Remember, this is the electron donor, NADPH. So in other words, if a cell is making fatty acids, it can take glucose and basically run through this set of reactions in order to get the NADPH it's used for fatty acid synthesis. So what does this, all of this depend on? Thiamin. Because what happens is when we get here, when these start recycling, it goes through trans-ketolase. And trans-ketolase uses thiamine pyrophosphate. Okay, now that's one way that it can go. This thing is also, it can turn in either direction essentially. So you can go from glucose 6 to fructose 6 and then go through this way. Either way.
Can you repeat why trans-ketolase is thiamine dependent?
It's its coenzyme.
Oh.
Now, here's one of the reasons this is really important for thiamine, say metabolism and assessment. Trans-ketolase is in a variety of tissues, including red blood cells. It's also in liver cells, quite a few other cells. So you can say trans-ketolase is in a variety of tissues, including red blood cells. Now, one of the reasons that's so important is, now what we can do is we can take a blood sample, isolate the red blood cells, and measure trans-ketolase activity. If it's low, what would we say about their thiamine status? It's probably inadequate or low. If trans-ketolase activity seems to be okay, it's an indicator their thiamine status is okay. One of the things that I mentioned, I said when we're trying to determine a recommendation for the population, it's very helpful to have a specific biochemical or some type of assay to measure that nutrient function. This is a functional assay that we can use to determine a person's thiamine status, and the recommendations in the US were partly based on this. Take individuals' red blood cells, they test it, and then that goes into how much thiamine we think they actually need. Okay? At the end of today's lecture, we'll go through a functional assay that has to do with this, so there'll be a little bit more detail on this. Okay? So if you look at this, theoretically, thiamine could affect DNA and RNA, right? So it could affect protein synthesis from RNA, it could affect cell replication. It can also affect the ability to make fatty acids and other molecules that are dependent on this. Okay? Now, the last kind of function of thiamine that we're going to look at.
Oh, sorry, I had a question on the last slide actually. So is trans-ketolase a reaction from the D-ribose phosphate, like those two groups, to back through the cycle?
Yeah. Trans-ketolase does this. So this molecule, yeah, see these little, I think there's molecules they left out here. Yeah. Trans-ketolase is required to go this way, and it's reversible. Okay, so the last part about functions of thiamine. So this is non-enzymatic. So there's a form of thiamine which is thiamine triphosphate. It's also thought to have important functions. But this form of thiamine is not functioning as a coenzyme. Thiamine triphosphate has important functions, but it doesn't work as a coenzyme. It's believed to be part of nerve cell membranes and involved in nerve impulse transmission. Okay, so let's see if I can summarize this quickly. It's believed to be involved in electrolyte gradients of plasma membranes, especially of nerve cell membranes. One of the reasons they think that's the case is, so it's somehow associated with the membrane, and when you take neurons in the lab and you stimulate them, the thiamine triphosphate hydrolyzes. The phosphates come off. When you stimulate neurons, the thiamine triphosphate hydrolyzes. So that's one of the reasons it's believed to be an important form for this. The other reason is when we talk about the physical manifestations in thiamine deficiency, the nervous system is one of the most affected systems in the body in thiamine deficiency, and it's thought this might be one of the reasons why. Okay, so the recommended intakes. We went through this. This was established in 1998. It hasn't changed since then. The Thiamine Committee set the DRI as an RDA, and these are the amounts. So this is the requirement. It's rather than 1.2, 1.1, it's 1 and 0.9. So what do we call this? Estimated average requirement. So this is EAR. So the way it's slightly less. For men, it's 1, slightly less than the RDA, because the RDA is 1.2, so we've added our 2 standard deviations. Okay? During pregnancy and lactation, the needs increase to 1.4, 1.5 milligrams per day. Kind of typical of a lot of nutrients.
So the RDA and DRI are what you should have, and the EAR is what you require.
So the EAR, remember, that's adequate amount for 1/2 of the population. Yeah. And the RDA should cover 98% of the population. Is thiamine deficiency common in the U.S.? What do you think? Why or why not? Enrichment program is a big part of it. Also, some of the sources of thiamine that are good sources, we tend to eat higher amounts in the U.S. than a lot of other places. So thiamine deficiency in the U.S. is not that common. However, the history of thiamine deficiency is significant. So if you read this that was written quite a while ago, 1630, by an individual in Java, this is what he wrote. So they called it Beriberi. And in this condition, they said, there's a certain very troublesome affliction which attacks men. It's called by the inhabitants Beriberi, which means sheep. I believe those whom the same disease attacks, with their knees shaking and legs raised up, walk like sheep. It is a kind of paralysis, or rather tremor, for it penetrates the motion and sensation of the hands and feet, indeed sometimes the whole body. Okay? So these individuals were having difficulty walking and moving. And kind of an inability or incoordination of movement is called ataxia. Have you heard that term? Ataxia is an incoordination in movement. Okay. Another condition that's related is called peripheral neuropathy. Peripheral neuropathy. And as the name implies, it tends to be dysfunction in the nervous system that starts in the peripheral tissues. In other words, fingers, toes, feet, hands, and then kind of works inward. So peripheral neuropathy. It tends to be symmetrical and affects both motor and sensory neurons. Sometimes you'll get tingling and numbness.
Can you repeat the definition one more time?
Sure. It is symmetrical. It's dysfunction of the neurons that involve sensory and motor neurons. So it affects coordination. It also affects sensory, like tingling, numbness. You'll see that there's quite a few nutrient deficiencies that affect this. Neurons are sometimes, depending on the function of the nutrient, one of the first types of tissues or cells to be affected. And one of the reasons for thiamine, I would say, is not only because of the thiamine triphosphate mechanism, but also neurons tend to be high energy requiring cells. Thiamine's really important for nerve conduction, especially in carbohydrate metabolism. So in the case for thiamine, I would say that's probably why the nervous system can be affected early on.
So wait, saying uncoordination due to thiamine deficiency, so that means their bones being affected as well, like topically there to the body, or?
It could, theoretically, yeah, because brain tissue is affected by thiamine deficiency. I don't know if it has to be.
Okay.
Yeah, and I don't think it has to be, because sometimes sometimes they'll do imaging on this, and I think you can have some of those symptoms without having any alterations in the brain. Mm-hmm. So this is what beriberi is. So I want you to think of beriberi as the classic thiamine deficiency. Okay, that's different than what we're going to say is kind of the other type of thiamine deficiency is more common in industrialized nations. Beriberi was really common in developing countries still. And some countries, for example, that relied heavily on rice for a big part of their diet, as soon as the processing of rice became popular, thiamine deficiency reached epidemic proportions because they were removing part of the rice kernel that had the thiamine, and they were not enriching the rice, and so tons of people in those populations became thiamine deficient. Okay, once they figured that out, now you can basically try to implement programs that protect against that. You know, we're going to talk about, however, some individuals consume alcohol, and that can complicate thiamine metabolism. Okay, so this is classic thiamine deficiency. Initial symptoms of beriberi may occur within a few weeks with no thiamine intake, and often the first symptoms include loss of appetite and weight loss. So clinical signs, I want you to remember these. Anorexia, the ones in red. Confusion, peripheral neuropathy. It's also associated with weight loss, apathy, loss of short-term memory, GI tract distress, irritability, muscle weakness. Seems to affect the nervous system preferentially, even though many tissues and processes are thiamine dependent. Why might this be the case? And I said, because of the role of TTP and thiamine's role in energy metabolism. Now I have here some of the clinical signs of beriberi can be seen in only 7 days on a thiamine-free diet. I'd say that's kind of exceptional, but I'd say definitely in a few weeks if a person's not consuming thiamine, it can be a problem.
What did you say to that question? Why might that be the case? Was it because of
TTP and its role in energy metabolism. Okay, there's 3 types of beriberi. We're going to talk about 2 of them. First one is dry beriberi. This usually means there's no cardiac involvement. It's likely due to chronic low consumption of thiamine, and it's characterized by muscle loss and weakness. The difference between dry beriberi and wet, one of them is the presence or lack of edema. In wet beriberi, there's edema. So it's likely associated with cardiac failure. And how would you define edema? It's fluid accumulation. Basically, it's interstitial fluid accumulation. And that could occur in several different parts of the body. In this one, it's typically extremities, mostly the feet. And you can see, for example, this probably healthcare worker has taken their thumb and depressed it into the person's foot, which is swollen with edema. This is where you don't want to hopefully end up, is on a nutrient deficiency slide in a nutrition class. You can see the foot is really swelled. This type of edema, it can get extremely hard. And there's so much tissue there, so much fluid there. When you depress it, it doesn't bounce back right away. So it takes a few minutes in order for that to go back to normal structure, which is significantly swollen because of the fluid. Okay? Mm-hmm.
If you had beriberi and were to increase your thiamine intake, would it theoretically go away?
Theoretically, yeah. Depends how long and how significant the deficiency was. Mm-hmm. Mm-hmm. This is the kind of type of thiamine deficiency that's more likely in industrialized countries. Wernicke-Korsakoff Syndrome tends to be something that more associated with alcohol intake. Now these are 2 physicians. Wernicke is one physician, Korsakoff is another one. The Korsakoff symptoms of this syndrome are more related to the mental aspects. Okay, so sometimes a person will have Wernicke's symptoms but not Korsakoff symptoms. Okay? So the point of this is you may have all the symptoms of the syndrome, a person may not. So sometimes it's also known as Wernicke's encephalopathy, which in that case typically means sometimes they're used interchangeably. So if you see that term, it's basically related to Wernicke-Korsakoff Syndrome. Okay, this is why alcoholics are at triple threat for thiamine deficiency. A lot of times they have a decreased intake of thiamine. A lot of times the alcohol is replacing thiamine food sources. Decreased absorption of thiamine. Alcohol decreases the release of thiamine from enterocytes into the portal vein. Alcohol decreases the release of thiamine from enterocytes into the portal vein.
So I know you said the Korsakoff's part is more related to what?
The mental aspects.
Decreases the release of thiamine from the enterocytes.
Into the portal vein. So if it doesn't get into the portal vein, it's not absorbed into the tissues, and that it can cause the thiamine deficiency. There's also the chronic intake of alcohol increases the requirements. One of the reasons is ethanol in the liver can decrease thiamine's conversion to coenzyme. So the presence of ethanol in liver cells can decrease the conversion of thiamine to coenzyme. The other thing I forgot to mention here, decreased absorption and increased excretion. So chronic alcohol tends to increase thiamine excretion. Okay, and remember we've said kind of in general, water-soluble vitamins not stored in significant amounts. So it's important that a person consistently takes in thiamine through their diet. Alcoholics, chronic alcoholics tend to not do that. So it's common for them to have thiamine deficiency. Manifestations of this are ocular dysfunction. So the eyes can be subjected to involuntary movement. A lot of times they'll kind of pulse back and forth. So ocular dysfunction represents involuntary movement of the eyes. Sometimes paralysis of ocular muscles. There's also ataxia. So their movement can be affected. So those tend to be the major symptoms for Wernicke's encephalopathy. If there's loss of recent memory and confusion, those are considered the Korsakoff syndrome symptoms. So in that case, the individual would have full-blown Wernicke-Korsakoff syndrome. Now, if you think about it, let's say you're in a hospital or healthcare situation and you encounter an individual that has alcohol on their breath, their eye movement is altered, their moving is affected, loss of recent memory, basically they're confused. What does that sound like? They're inebriated. Okay? So healthcare professionals are basically aware that it could be inebriated and or could have thiamine deficiency. Commonly they overlap. it's not uncommon for them to give that individual a thiamine injection just to make sure. Because if it's not diagnosed and that person remains thiamine deficient, it can cause permanent damage to their brain. Okay? If there are brain lesions, they can be irreversible. So here's an example of a water-soluble nutrient deficiency causing irreversible long-term problems. Now, and I've seen some of the images of the brain. It's pretty drastic on some of these, and again, it depends how significant it is and how reversible it is. But it's important that that does not continue the thiamine deficiency. Now, not everyone, not all alcoholics have these symptoms. So it's believed there's a genetic component. It's thought maybe that it has to do with trans-ketolase, and that some individuals have a genetic alteration in trans-ketolase structure that kind of sets them up for manifesting Wernicke-Korsakoff Syndrome. Mm-hmm.
So even if you eat enough thiamine during the day, or even more than required, you still have a disposition to drink a lot more. You wouldn't have these symptoms of Wernicke.
It means that if a person's thiamine deficient and part of it is being caused by alcohol intake, some people kind of are protected against that depending on the trans-ketolase structure, yeah. Some people are more sensitive to it, I guess. Okay? That's still, I would say, debatable. A couple papers hypothesizing that, not a real ton of data on that one. Treatment. What do you do for Wernicke-Korsakoff Syndrome? Therapeutic levels of thiamine, 100 milligrams. Is that a lot? Yeah, the RDA is about 1 milligram. That's 100-fold. Why isn't it harmful? There's no UL. Okay? So typically it can be oral, but in this case, usually it's an injection of thiamine. UL for thiamine, there isn't one. Therapeutic levels for inborn errors of metabolism, right? Like maple syrup urine disease. Sometimes they are given up to 500 milligrams a day. Remember I said some individuals with maple syrup urine disease are what we call thiamine responsive. Their symptoms will improve if we give them significant amounts of thiamine. Sometimes it's 500 milligrams. So in other words, sometimes almost 500-fold the RDA for that. Now, how do we assess thiamine status? There's a few ways. One is you can actually measure thiamine in the blood or the urine. And there's certain ranges in both of those components that you would determine whether or not they're in a normal range of thiamine. Another way of doing it is doing an erythrocyte trans-ketolase activity assay. So this is a functional assay that's sometimes done, and this is how we do it. So if I ask you how to use a functional assay to assess thiamine status, this is basically what you'll explain. And you'll probably have a diagram or a drawing in there. Okay? So you obtain a blood sample. Okay? You add a buffer to lyse the red blood cells. Why do you do that? Because the trans-ketolase is inside the red blood cells. So you open the red blood cells, then you measure trans-ketolase activity. Then you add thiamine pyrophosphate. Incubate for 30 minutes. Measure trans-ketolase activity again. If that person is thiamine deficient, would you expect the second trans-ketolase activity to be higher or lower? Who says higher? Who says lower? It's 3-0. Great voter turnout. 3 people voted.
Can you not get lower?
Probably not, but theoretically it could. But what if it's the same? Okay. Now think about what's happening. Trans-Ketolase relies on thiamine pyrophosphate, right? So again, let's say we had the scenario where in their red blood cells, here's trans-ketolase. There's a few copies of it. Let's say in this individual, they have a few copies of trans-ketolase. Only one of them has thiamine pyrophosphate attached. Measure the activity of all the trans-ketolase in the red blood cells, get a value. Now we add more TPP. What happens to overall trans-ketolase activity? Goes up. Why? Because now you're providing more coenzyme for the enzyme.
So if there's no change in your trans-ketolase activity by the end of it, does that mean you aren't thiamine deficient?
You're likely adequate thiamine status. Yeah. You're not benefiting from the addition because you already have adequate amounts of TPP to saturate the binding sites. Okay? So an increase in trans-ketolase activity of greater than 25% indicates thiamine deficiency. An increase of more than 25% in activity indicates deficiency. Got it? If it's no increase or less than a 15% increase, person's considered adequate. Got it? Questions on thiamine? Okay. Let's go to riboflavin. Okay, riboflavin. It's vitamin B2. You want to remember it's vitamin B2. All right, water soluble. Here's riboflavin structure. It has I'd say two parts to it. Riboflavin consists of flavin and ribitol. So here are the flavin ring structures. Here's ribitol. Several alcohol groups here. These two are attached. Flavin is from Latin with flavus, which means for yellow. Flavin gives the vitamin its yellow fluorescent characteristic color, right? You can see that color sometimes in a person's urine if they've taken a relatively significant amount of thiamine. You also notice it when I was in grad school, we used to make up diets for our lab animals from all the separate components, and we were told it was a really good way to learn about the different components. Later on I found out it was just because my boss was really cheap. And it was cheaper for us to do it that way. So we take each of the vitamins. Sometimes we take a vitamin mix and add them into our rat diets, mouse diets, whatever we were making. And the advantage of that was if we then wanted to induce, for example, a copper deficiency, an iron deficiency, or a zinc deficiency in these animals, we would just leave out that nutrient. And basically, we had a way of studying what zinc deficiency, for example, does to individuals. In that case, rats, but obviously a lot of it extrapolates. One of the things you notice about riboflavin is it's got that really distinct, not only yellow color, but the smell. Have you ever tasted anything or smelled anything? You're like, oh, it tastes like vitamins. How do you know what vitamins taste like? You just kind of sometimes associate, oh, that's got a lot of vitamins in it. That's got a kind of an odd taste. A lot of times that's riboflavin. Sometimes that's the one you have to cover up if you're trying to make a product that has vitamins in there, including riboflavin. But you can definitely smell it. I remember opening that jar, and riboflavin fills the room, and it was kind of almost a pasty kind of gel kind of stuff that we would add in there. It's always in a brown bottle. Why is it in a brown bottle? Yeah, it's sensitive to UV light. What's another example of protecting riboflavin, something that we buy a lot? Milk. It's one of the reasons milk is in cardboard, opaque containers is to protect the riboflavin. Years ago, and you guys don't remember this, I remember when I was a kid, we had a milkman. Saturday morning, that milkman would come around. They'd actually put the milk in a glass bottle outside of our patio, and I used to love it because one of those half gallons was chocolate milk. By Saturday night, man, most of that chocolate milk was gone. I'd hit that hard. But they know one of the reasons they put milk in different containers now is to protect the riboflavin. Because of those ring structures, UV light damages riboflavin significantly. It's relatively stable to heat, but not UV light. Okay, so it's one of the ones that we want to protect.
How is the UV light getting into a refrigerator?
Oh, it's mostly during transport and storage, yeah. And the glass will protect a lot of it. Not a lot of UV goes through glass, but some of it can. Yeah. What else? I think that's good. Well, so what does riboflavin do? Well, riboflavin is the vitamin. It's got two coenzyme forms. So riboflavin is converted to Flavin Mononucleotide. You ever seen FMN anywhere? That's probably the least famous of the coenzyme forms, but it is a coenzyme form. And if you look at what it is, here's our flavin rings. Here's ribitol. This ribitol, though, now has a phosphate group. This is considered a nucleotide. So it's Flavin Mononucleotide. So phosphate's been added. More on that in a minute. The other coenzyme form is the more, let's say, popular one or famous one, Flavin Adenine Dinucleotide. In this case, the addition's more complex. Here's the phosphate from mononucleotide. Now we have another phosphate that's added. So this is from pyrophosphate. Now this forms pyrophosphate. Here is adenine, and here's a ribose that attaches it. So here's our adenine dinucleotide that's attached to the vitamin. Right, and we know we're familiar with FAD. We'll talk about that more. I'll show you the reaction in a few minutes. Dietary sources. Which are the best sources? So it's found in a wide variety of foods, riboflavin. It tends to be higher in animal products. In the U.S., milk products, basically let's say dairy products, are the most significant contributor to the diet. In the U.S., dairy products tend to be the most significant contributor. Okay, now you'll find both the vitamin form and the coenzymes in food. So foods can contain the vitamin and coenzyme forms. You can get riboflavin, FMN, FAD. Which of those contribute to our riboflavin intake? In other words, which do we count? All of them. So similar to thiamine and thiamine pyrophosphate, we can consume FAD, convert it to riboflavin, and then back to coenzyme. It's kind of deja vu. In general, animal products have more FAD than riboflavin and FMN. So FAD tends to be higher. In most foods than the other forms. Now, milk has a higher riboflavin to coenzyme ratio than most other foods. But other significant sources of riboflavin include eggs, and enriched and fortified cereals. Okay? So I'm going to give you 3 sources with all that said. These are the riboflavin sources to remember. Number 1, dairy products. Second one, grains. Which kind? Whole, enriched, fortified. And the last one, meats. Meats, a variety of different types of meats.
So you said that most food products have more of the coenzyme than other forms, but except for milk, it has higher riboflavin?
It has a higher ratio. It doesn't mean it has
Doesn't mean it has
more.
more.
Yeah, it means that compared to other foods, it has more riboflavin compared to their amounts.
Okay, got it.
Okay, so if we look at this. Overall, riboflavin is relatively widespread across a variety of different foods, and you can see here, you can see how the dairy products are kind of standing out here, significant amounts. This also indicates the DRI is an RDA. For men, the RDA is 1.3 milligrams a day. For women, 1.1. What is that relative to thiamine? Just a little bit more, probably about 10% more. So these two nutrients are relatively similar in their amounts for their recommendations. Okay, now what do we do? Well, the diet contains riboflavin, FAD, and FMN. If we consume the coenzyme forms, there are enzymes in our gut, specifically pyrophosphatases and phosphatases, that convert FAD to FMN. Right? This is cleaving in between the phosphates. So we go from FAD to FMN, and then that final phosphate's removed to give us riboflavin. That's how it's absorbed. So again, coenzymes are consumed. They're modified to vitamin to be absorbed. This occurs in the lumen of the small intestine. So like thiamine, most digestion, absorption occurs in the small intestine. In intestinal cells, absorption is by a saturable, energy-dependent process. So in other words, there's riboflavin specific transporters that are on the apical membrane. Okay? Even though it's energy dependent, it's not sodium dependent. About 95% of riboflavin from foods is absorbed up to a max of 25 milligrams. And maybe I worded this incorrectly. Because even if you consume coenzyme form, 95% of that is going to be absorbed as riboflavin. So in other words, the conversion of coenzymes to vitamin is not rate limiting. Okay? This is up to a max about 25 milligrams. Is that a lot? Yeah, that's over 20-fold the RDA. Then what happens? Diffusion becomes more significant. So once the active transporter becomes saturated, diffusion is also possible. After the active transporter is saturated, diffusion becomes more significant. Now, once the riboflavin goes inside the enterocytes, and other cells for that matter, it can be converted to coenzyme forms. So the first enzyme that's involved in this process is an enzyme called flavokinase. It converts riboflavin to FMN. It uses a phosphate from ATP. And that's it. Then you have FMN. FMN can function as a coenzyme, or it can be converted to FAD. Right? Now, I mentioned this with thiamine. Before exiting a cell, FMN is converted back to riboflavin for excretion into the portal blood. So same principle. Vitamins go across membranes. Coenzymes tend not to. So vitamins go across cell membranes. Vitamins tend not to. What's that?
Vitamins go across
Cell membranes.
and
Coenzymes tend not to. So here's the first reaction. Riboflavin go into a cell, converted to FMN. Then FMN, right here, Flavin Mononucleotide, can be converted to FAD. The enzyme that converts FMN to FAD is called FAD synthetase. That makes sense, right? Now this reaction is a little bit different because rather than just removing a phosphate, you need the adenine. Because now we're making adenine dinucleotide. So if you have ATP and you take the adenine and a phosphate and separate it from two phosphates, that's what's added to FMN. So your reaction here results in two inorganic phosphates. So what are you adding? AMP essentially. Got that? Now, almost any cell in the body can do this. It can go from riboflavin to FMN to FAD. Because FMN and FAD are so important for coenzymes, lots of cells can convert them. You guys know one example of what's FAD needed for? Energy, more specifically. It's used to contribute electrons to ETC. Remember beta oxidation of fatty acids. What else? Krebs cycle. So very important for energy. FMN, same thing. It's used in ETC. We'll look at that in a second. So my point is, Really important in energy metabolism, so most cells can go from riboflavin to these coenzymes. Now, if a cell releases riboflavin into the blood, it's usually kind of bound to proteins as it does that. So in the blood, it's mostly riboflavin, not coenzyme. And it's typically bound to albumin, fibrinogen, or antibodies. So one of the points to this is there's not really a riboflavin specific protein in the blood that's function is to transport it around. But riboflavin can kind of loosely associate with this protein, these proteins. Okay. Then riboflavin can go into cells and function. Now, one of the reasons riboflavin is an important part of coenzymes is because of the structure of those flavin rings. That structure and these nitrogens support the addition and release of hydrogens, right? Basically, hydrogens can attach and dissociate from these nitrogens. So if we're looking at FAD, for example, we have this ring structure that can be in its oxidized form, which is FAD. If it's reduced, we call it FADH2, right? So here's the ribitol here. And again, imagine that this is attached to an enzyme. And the enzyme is using the FAD or the FMN to gain and transport electrons. That's what it's using it for. So it's very similar to this. Right? So in this, rather than TPP, it would be FMN or it would be FAD. Same basic principle.
Transport protons?
Electrons. Okay? Now, obviously these are different enzymes than those that use thiamine. And there are lots of enzymes that require flavin coenzymes, FMN and FAD. It's estimated there's over 200 enzymes. Over 200 enzymes use either FMN or FAD. Sorry, up to 200 enzymes. Here's a few examples. We mentioned this before. Electron transport chain. More detail coming up. Succinate dehydrogenase, which is in Krebs cycle. Remember that one reaction in Krebs cycle that goes FAD to FADH2? Also, look at this one. We can synthesize niacin from tryptophan. What kind of molecule is niacin? A vitamin. How about tryptophan? An amino acid. So the next vitamin we're going to talk about, we can actually make some from tryptophan. Riboflavin is required for that. So if a person is inadequate in riboflavin, they could also be inadequate in niacin because they can't make it anymore. Synthesis of active coenzyme form of vitamin B6, which is also known as pyridoxal phosphate. So riboflavin-dependent enzymes help us make two other vitamins. Fatty acid oxidation we went through, and the list goes on and on. Again, there's somewhere between 100 and 200 different enzymes that are part of this process. This is kind of a summary. I think I got this from a 210 text on different reactions requiring different nutrients. So any place on here you see FAD or FMN, it's riboflavin dependent. All right, so if we go through here, for example, here's FAD, and this is supposed to represent what? Here's fat going to acetyl-CoA. Here's beta oxidation. Down here, here's our succinate reaction. Here is electron transport chain. There's two places riboflavin is required. FMN is part of one of the ETC components, and FAD can contribute electrons here. There's a few other examples that aren't up here. So here's one way of looking at ETC, and this is in, I think, chapter 1 of your textbook. Looks kind of messy. Where are we? Here's mitochondrial intermembrane. Here are components of the ETC. So down here is the last one. So here's complex 4, cytochrome oxidase, that's adding electrons to oxygen and protons to get water. So if we backtrack here, here's where we say, remember FADH2 that's generated generally from Krebs can contribute electrons to right here, complex 2, coenzyme Q. That's what's happening there. We'll also look at in a few weeks how iron's involved in that. Basically, the electrons go through iron to get here. So there's the FADH2. Here's FMN. And I didn't mention this before in 328, but complex 1, we called NADH dehydrogenase. It actually uses an FMN coenzyme to take the electrons from NADH. The electrons bind, and then they're transported through an iron-sulfur cluster, eventually to coenzyme Q. So what you can say is FADH2, FAD is required for coenzyme Q, and FMN is required for complex 1 of ETC. All right, so easy to see if a person's riboflavin deficient, their energy metabolism is significantly compromised. So here's PDH. Very seldom when we have a lecture in here we don't talk about PDH, it seems like. This is pyruvate to acetyl-CoA. Now we're used to just going, oh, pyruvate to acetyl-CoA generates NADH. There's a previous step where FAD is involved. Remember this from that slide that you on the pyruvate decarboxylase and PDH with thiamine? Here's our FAD. So this conversion, pyruvate to acetyl-CoA, also riboflavin dependent. So pyruvate to acetyl-CoA is also riboflavin, specifically FAD dependent. Here's Krebs cycle. Succinate to fumarate requires FAD. Does it look a little different now? Because I think before when you were doing Krebs cycle, you're just going, oh, this is what's made. Why is it made? Because the enzyme succinate dehydrogenase is using this as a coenzyme. So these aren't just floating around. They're coenzymes. So Krebs cycle activity can be compromised in riboflavin deficiency. Fatty acid oxidation can be compromised. So you guys had this in 328 as well. The first oxidation step of beta oxidation is FAD dependent.
Should we remember these specific steps that, like, or how would you ask, you just need to know that it's beta oxidation that you is dependent on it, or should we remember the step that
In this case, you could just say, you know, the enzyme in the first oxidation step of beta oxidation requires FAD. Yeah. So point is, we can see, I think, if you don't have riboflavin in adequate amounts, significant manifestations or symptoms. Now for riboflavin, metabolism and excretion. Riboflavin and its metabolites are excreted in the urine. Most riboflavin is excreted intact in the urine, about 60 to 70% of it. And that's why it can appear in the urine as kind of the colored urine in a couple hours. So the typical amounts found in vitamins can change the color of the urine to a bright yellow color. That's what a lot of people will see. The reason you see that is the riboflavin wasn't broken down. Those rings are still intact, and they're providing that yellow color. So it's a little bit different than thiamine. Thiamine is more metabolized before it's excreted. Riboflavin is more excreted before it's broken down. The RDAs. The requirements for riboflavin intake have been estimated on studies involving intake and urinary excretion, so they kind of compare those. Intake levels and clinical signs of deficiency. We'll look at those in a second. And an erythrocyte, glutathione reductase activity. So this is an assay. What kind of an assay would we call it? It's a functional assay. Okay. So there's significant enough data and a specific test we can use to make the DRI an RDA. If you have an RDA, you have an EAR. EAR is always less than the RDA. The EAR is 1.1 and 0.9 for males and females, less than the RDA, which is 1.3 and 1.1. So, deficiency.
What is a functional assay?
That means you are using like a functional test to assess a person's, in this case, nutrient status. Yeah, you're measuring the functional activity of an enzyme in that case. Okay? Deficiency of riboflavin is called Ariboflavinosis, and it rarely occurs without other deficiencies also being present. No clear riboflavin deficiency disease has been characterized, and all that means is most of the time if you have a riboflavin deficiency, it overlaps with other deficiencies. Now, clinical signs appear within 2 to 4 months of inadequate intake. Signs of a deficiency in riboflavin include chelosis, which are lesions. Lesions is the best term. Yeah, lesions on the outside of the lips is chelosis. Angular stomatitis, have you ever heard that? That's the lesions at the corners of the mouth. What's that?
Sorry.
Angular.
Cheilitis was at the corner, or cheilitis at the corner of your lips.
Chelosis is overall. If it's at the corners, you can say it's angular chelosis or stomatitis.
So they're interchangeable.
Next one is glossitis, which is inflammation of the tongue. That's what's shown there. Seborrheic dermatitis, inflammation of the skin. Anemia, we know. Peripheral neuropathy, we know. So if I ask you to tell me, basically list, let's say, 3 symptoms of a riboflavin or signs of a riboflavin deficiency, and why riboflavin deficiency is causing those, one way to look at it would be you can kind of associate an energy deficiency with a lot of these. So for example, these involve epithelial cells, and epithelial cells, like neurons, require significant energy. Uh-huh.
So we could list, if you asked us that question, we could say, we could pick 3 of those things, and then for why the deficiency is causing it, it would just be because all of those cells require epithelial cells, which require energy. That could be the answer.
Those tissues are showing deficiency because of compromised epithelial cells.
Okay.
Skin on that one as well. Neurons we know is energy dependent. Anemia is different, has to do with protein synthesis. Okay, that's probably a good place to stop. Any questions? Okay, so today I'll send you guys, I'll post the thiamine study guide, and then I'll convert this one as soon as I can to YouTube and put it up. I'll send you an email when it's converted with the link. And I'm missing anything? Uh-huh.
So is it on the part of riboflavin that we just did, or just thiamine?
Thiamine and all the, through riboflavin up to here, yeah. Okay, thank you guys.